Tertiary structure is the final folded shape of a protein chain. And that shape is stabilized by a number of forces, sometimes including sulfhydryl bonds, covalent bonds between cysteine side chains. Hydrogen bonding can be very important. Ionic bonding between charged side chains of different amino acids can stabilize protein structure. Now, when we talk about tertiary structure, we're only talking about a single chain, not multiple chains. So we're only talking about a single chain. And if this, imagine this is the shape that the protein folded into, that we could refer to as the tertiary structure. But some proteins consist of more than one polypeptide chains. So quaternary structure is the final level of protein structure, and it is the arrangement of individual chains that we'll call subunits in a protein that has two or more polypeptide chains. So if you have a protein that has two subunits, let's say, there's one protein chain, and that comes together with another protein chain. And this now is the functional protein. This would be a dimer because there are two subunits. There are two polypeptide subunits. And we would refer to the overall structure of these two polypeptide chains coming together. That would be the quaternary structure. So quaternary structure only applies when a protein has more than one subunit. Now you can have homodimers for proteins, and those would be where the two subunits are identical. Homo meaning the same. So an A subunit comes and interacts with another A subunit, and this forms the functional protein. But there are heterodimers as well. And that would be where two different protein chains came together, let's say A and B, to form a functional protein. This would still have quaternary structure, but this would be, the quaternary structure would be formed by two different polypeptide chains. And of course you can have trimers and multimers that go beyond just two. I just use that as an example. All right, so here's a diagram of some of this. Here's the primary structure. Here is an alpha helix secondary structure. Here is the tertiary structure of a single polypeptide chain, a single protein, and it has an amino end and a carboxy end because at the beginning of the protein you have a free amino group for the first amino acid, and at the end of the protein you have a free carboxyl group because it's not there's no more amino acids in the chain. So there is a polarity to a polypeptide chain. And then quaternary structure is when you have more than one polypeptide chain coming together, and these then form the subunits of the entire protein. And here are some more diagrams. Primary structure, the sequence of amino acids. Here's a beta sheet, and we're showing the hydrogen bonds now that link one beta strand and another beta strand, and they are forming between their hydrogen bonds are forming between the carbonyl carbon on the main chain and on one strand, and the amino nitrogen and hydrogen on the other strand. So this is a case where this nitrogen is sharing this hydrogen with this partially negatively charged oxygen. That is, this forms the classic definition of a hydrogen bond. Likewise, alpha helices are stabilized by hydrogen bonds occurring between the same carbonyl carbons and the amino nitrogens and hydrogens. They share this hydrogen here. And so what we have along a helix is a series of hydrogen bonded participants that stabilize this twisted helix shape. Tertiary structure would be the overall shape of a single polypeptide chain. And so the tertiary structure might have beta sheets in it, alpha helices, loops that connect the various secondary structure elements. These here are loops. But the overall shape of the protein then, of the protein chain, is referred to as tertiary structure. And finally, quaternary structure is when you have more than one protein subunit coming together to form a functional protein. Now, motifs are common elements of secondary structure that are shared among families of proteins. So a particular motif that I've talked about already in this class is that the homeodomain motif. And that is several elements of secondary structure come together to form that motif. And that structural motif is found in a family of proteins. And if you find a motif like that that's found in known proteins, but you're studying an unknown protein, you can get a pretty good idea of the function of that protein based on the known function of those motifs in other proteins. And domains are functional units within a larger structure. So let's imagine that you had a protein chain like this, a protein shape that's something like this, and this pocket right here, color it in, might be a binding site for chemicals that with the enzyme, if this is a protein enzyme, the enzyme would take the reactants and catalyze the formation of products. Whereas, let's say, this part of the protein over here might be involved in interacting with another protein. And so what we would have in our original protein then are two major domains that are different, have different functions in a way. So they're functional units within a larger structure. And most proteins, in fact, do have multiple domains that perform different functions for and endow the protein with its overall function. So here are some motifs. Here's a beta-alpha-beta motif, beta sheet connected to an alpha helix connected to another beta sheet. Helix turn helix motif, that is found in many DNA binding proteins, including the homeodomain that I mentioned. And then domains can be represented by functional units. This one might have one function. This domain could serve as a connector function between the domain 1 and domain 3. So it might be a domain, but its only function might be to connect to other domains. So domains are functional units within a protein. We'll have one more part to this lecture, a short part. We'll finish up and we'll look at the phenomenon of how it is that proteins fold, protein folding. And this is very important because, as we've seen, the three-dimensional shape of a protein is determinative of its function. That's where we'll go to finish up.